Hello everyone, welcome back to another episode of Doctor's Quiz. What we are going to see today is MCQs from microbiology from the chapter Bacterial Genetics. And if you are new to my channel, please subscribe to Doctor's Quiz to learn medical subjects through quizzes. Are you ready? Let's get started. The first question is, the codon created by which mutation prematurely terminates the growing peptide chain and destroys the function of the proteins? The options are silent mutation, missense mutation, nonsense mutation, and frame shift mutation. The right answer is nonsense mutation. The next question is, which test is used to test whether a particular substance can induce mutations or not? The options are microarray test, gene expression test, chromosomal test, and Ames test. The right answer is Ames test. The next question is, the process of gene transfer in which a bacterium takes up and incorporates free DNA from its surroundings into its own genome is known as? The options are transformation, transduction, conjugation, and fusion. The right answer is transformation. Next question is, the type of gene transfer in which some of the bacteria resist transformation until they are subjected to some special treatment to make them permeable to DNA. The options are conjugation, transduction, artificial transformation, and conversion. The correct answer is artificial transformation. The next question is, the type of gene transfer in which the DNA of one bacterial cell is introduced into another bacterial cell by viral infection is known as? The options are transformation, conjugation, conversion, and transduction. And the right answer is transduction. The next question is, how many types of transduction in gene transfer? The options are 2, 3, 4, and 5. And the right answer is 2. The next question is, a bacteriophage genome that is integrated into the circular bacterial chromosome or exists as an extra chromosomal plasmid within the bacterial cell is known as? The options are lytic phage, temperate phage, prophage, and none of the above. The right answer is prophage. The next question is, if due to any reason the phage DNA fails to integrate into the host chromosome, the process is called as? The options are co-transduction, abortive transduction, conjugation, and mutation. And the right answer is abortive transduction. The next question is, the transfer of more than one gene at a time is limited to linked bacterial genes. The options are generalized transduction, transformation, co-transduction, and specialized transduction. The right answer here is co-transduction. The next question is, the existence of prophage in a cell for a long period of time suggests a mechanism for the virology of? The options are cancer, hepatitis B, AIDS, and syphilis. The right answer is cancer. The next question is, the bacteria harboring the prophage in lysogenic conversion is? The options are lysogenic bacteria, lytic bacteria, phagocytic bacteria, and all the above. The right answer is lysogenic bacteria. Next question for you is the transfer of DNA directly from one bacterial cell to another by a mechanism that requires cell-to-cell contact. The options are conversion, transduction, replication, and conjugation. The right answer here is conjugation. The next question for you is the technique in which DNA is manipulated artificially to identify and derive useful genes and genetic products is known as. The options are DNA cloning, genetic engineering, DNA amplification, and genetic mapping. The right answer is genetic engineering. The next question is the process of introduction of gene and its multiplication in a host is called as. The options are cloning, mapping, replication, and coding. The right answer is cloning. The next question is the duplication of a gene isolated from another organism by the cells of a microbial host is known as. The options are genetic material, genetic transformation, genetic code, and gene clone. The right answer is gene clone, which has been traditionally considered as an ideal cloning host. The options are Saccharomyces cerevisiae, Bacillus subtilis, Escherichia coli, and all the above. Here the right answer is Escherichia coli. The next question is vectors used in cloning technique is of how many types? The options are 1, 2, 3, and 4. The right answer is 3. The next question is the plasmid that carries genetic markers for resistances. The options are R factor, Charon phage factor, hybrid vector, and none of the above. The right answer is R factor. Human insulin is synthesized by using which technology? The options are gene knockout, next generation sequencing, recombinant DNA technology, and gene cloning. The right answer is recombinant DNA technology. The next question is polymerase chain reaction is of how many stages? The options are 1, 2, 3, and 4. And the right answer is 3. The next question is Northern blotting technique is used for the analysis of. The options are DNA, RNA, proteins, and enzymes. The right answer is RNA. A technique that utilizes DNA hybridization to detect specific DNA sequences within a complex mixture of DNA is. The options are Southern blotting, Northern blotting, Western blotting, and Eastern blotting. Here the right answer is Southern blotting. Which blotting technique have been used for the confirmation of HIV antibodies? The options are Southern blotting, Northern blotting, Western blotting, and Eastern blotting. Here the right answer is Western blotting. The next question is Western blotting technique is used for the identification of. The options are DNA, RNA, proteins, and all the above. The right answer is proteins. The final question is, which has been used for the detection of Mycobacterium tuberculosis, Borrelia burgdorferi, and Neisseria gonorrhea? The options are blotting techniques, polymerase chain reaction, ligase chain reaction, and recombinant DNA technique. Here, the right answer is ligase chain reaction. Yes friends, that's all for today's video. Hope you have learned few MCQs today and let me know your scores in the comment section. I will meet you in another episode of Doctor's Quiz on coming Friday. Until then, bye bye. Take care.